Okay, and so up next we have Professor Anna Moore. She's at Michigan State University. She just recently joined us. She's a professor of radiology and physiology, and right now she's the director of our Precision Health group. And she's going to talk about molecular imaging, theranostic agents, and precision medicine in cancer therapy. Thank you.
Thank you so much. Thank you for inviting me to talk at this prestigious meeting. As you just heard, I just joined MSU a few months ago. I spent most of my professional career at Massachusetts General Hospital and Harvard Medical School, 27 years. And the results that I'm going to show today are primarily coming from that research. So I'm here to establish and develop a precision health program. But today I'm going to talk about a building block of that program, which is precision nanomedicine. So this is just my disclosure of my financial interests. And in the next slide, let's just talk a little bit about precision medicine. So the idea to use patient genome information is not actually new, and the term pharmacogenomics was actually coined in 1959. But it's only now we can bring this knowledge from niche to mainstream. And the reason for that is because the cost of sequencing a genome dropped dramatically, and I have a data for 2015. Now it's probably even cheaper. Also, we need to recognize the initiative of 2015, the White House initiative of precision medicine, where the first $200 million were pooled and given to scientists to develop that field. So the idea is basically to customize care based on the genotype and phenotype of the patient. So what we think will happen is that patient's genetic information will become part of his or her medical records, permanent part, and would be used by clinicians and your primary care doctors in maintaining your health and treating your disease. So basically, what does it look? Well, how does medicine look without precision medicine? So let's say we have a bunch of patients with lung cancer, and all these patients receive same therapy. And interestingly, the results are going to be different in these patients. Some patients will benefit from this therapy. Then there's going to be a majority, actually, of patients that are not going to benefit from this therapy. And then there's going to be a group of patients that are going to have adverse effects so severe that it's going to be hard to tell whether they benefit or not. So why this is all happening? Because all tumors are different. And for example, lung cancer can have up to 140 different mutations in smoker compared to non-smoker. So clearly, what we really need to do is to treat those patients taking into account genetic makeup of their tumors. So if we use precision medicine approach, then the treatment would look different. So we would have these patients, we would test their tumors' genetic makeup, and we'll give those patients drugs depending on how these tumors react to those drugs, whether they're going to be sensitive or not, and that would depend on the genetic makeup of those tumors. So clearly, there are a lot of elements that are dysregulated in cancer that we need to target as a scientists, as researchers. And I want to focus on one of them, which is called non-coding RNAs. Those are small RNA molecules that do not code any proteins. They're called microRNAs, and they affect our genome dramatically. So this is basically a slide that describes how microRNA work in very short paragraph. So we know now that up to 60% of our genome is regulated by those microRNAs, and they basically, what they do, they bind to mRNAs, and they inhibit genetic activity. They block the synthesis of the proteins. So microRNAs are dysregulated in cancer. They can be upregulated. They can be downregulated. And the idea behind therapy could be based on normalizing microRNA profile of tumor cells. So if we have a situation when microRNA is oops, it turned out that you can't press this button too hard. So, okay, I just learned something. So if microRNAs are downregulated, which means there are not enough of them in the cell, what we need to do is to add microRNA mimics, double-stranded RNA molecules. In that case, I hope I'm pressing the right button here. No? Oh, this one, the right. Sorry. So in this case, protein expression will be decreased because mRNA will be blocked by mimics microRNA. Now, if you have a situation when microRNA is upregulated, so what you need to do is to inhibit it. How do you do that? You would add microRNA inhibitors, complementary small RNA molecules. So those molecules would bind to microRNA that's upregulated, and as a result, you'll see the increased protein expression. So either way, whether microRNAs are upregulated or downregulated, you need to deliver small RNA molecules into the cell. And that presents a problem in the delivery. Why? It's very difficult to deliver small RNA molecule in the cell. One reason is because those molecules are prone to digestion by nucleases. They have a very short half-life, and they degraded in serum. RNA molecules are negatively charged, so they cannot cross cell membranes by themselves. And then we have an issue of target effects and activation of innate immune response. And again, that made delivery of active RNA molecule to the cell very difficult. So this slide basically lists all attempts that have been done in the entire world to deliver those RNA molecules in the cell. Various carriers, various polymers, various lipid nanoparticles, various nanodelivery systems been tried, and the effect has been very dismal, I would say. There's only few clinical trials. If you look at clinicaltrials.gov, and those clinical trials are not very effective either. And this is, again, because of the delivery issue. So to overcome the issue of the delivery of small RNA molecule, we introduce a delivery system, a modular platform that consists of a nanocarrier that incorporates therapeutic, imaging, and targeting components. So this nanocarrier protects RNA molecule from degradation and allows for non-invasive monitoring of its delivery to the tissue or to the organ. So therapeutic component of this molecule would be RNA, small RNA molecule. The imaging component that helps to monitor the delivery or monitor the outcome could be a fluorescent dye, could be PET isotope, or could be magnetic nanoparticles that are used in MR imaging, and those are actually the ones that we use, and I'm going to show you on the next slide. We can also incorporate targeting moieties. We can incorporate targeting peptides or other affinity moieties that would help those complexes to accumulate inside the tumor cells. So, and basically the beauty of this design is that it's a very flexible design. You can incorporate any molecules, of course, taking into account available chemistry, and deliver them to the cells. So like I just mentioned, as a delivery molecules, we chose to use iron oxide nanoparticles. This is sort of a historical decision because my professional life started with using magnetic nanoparticles for early cancer detection by magnetic resonance imaging. And we reasoned that if we can use those nanoparticles for imaging tumors, then we can probably use them as carriers to deliver drugs to those tumor cells. And in this case, our drugs were small RNA molecules. So this is basically the complex that we synthesized. So this is my interpretation of iron oxide molecule covered with dextran. It's a sugar that basically protects magnetic nanoparticles from falling apart. And then this dextran code is modified with amino groups. And there's a lot of chemists in this audience who knows that if you have an amino group, you can conjugate just about anything. And in this case, we conjugated 5.5 dye, which is a near-infrared fluorescent dye for correlative optical imaging and fluorescence microscopy. And also, we conjugated small RNA molecules, oligonucleotides, targeting a specific microRNA, microRNA 10B, and I will tell you in the next slide why we chose that one specifically to target. We also synthesized the molecule that carrying scrambled RNA molecule for control studies. So the choice of microRNA 10B was based on the data that actually Sophie showed in her presentation as well. We're showing the same table of a dismal survival of patients with breast cancer. 5-year survival is about 20%, and there were over 250,000 cases in 2017, and I don't have the data for 2018, but I don't think it's getting any better. So we chose breast cancer as our first indication, but as I tell you later, this approach could be applicable for other metastatic cancers. So we targeted microRNA 10B, and microRNA 10B was discovered in the lab of Bob Weinberg at Harvard. And what they showed was that this microRNA was highly expressed in metastatic cancer cells, and it regulates cell migration and invasion. Its role in metastasis was also clinically validated. It was showed that it was overexpressed in metastatic tumors compared to primary tumors, and then it was overexpressed in primary tumors with patients that went on to developing metastasis compared to patients that did not develop metastasis. So the idea was to use our nanodelivery system to deliver oligonucleotides inhibiting microRNA 10B with two purposes in mind. One was to prevent metastasis from spreading from primary tumors, and the second was to arrest the growth of already formed metastasis. So the add-on value of this system, as I mentioned before, was the ability to image the accumulation of this nanoparticle carrying oligonucleotides non-invasively. So in this next experiment, we first proved that we can inject nanoparticles and they would accumulate in the primary tumors and in metastatic sites. So here I'm showing the results of MRI imaging showing pre-injection T2 map with the primary tumor right here. And this is a post-injection image that's showing a decrease of signal intensity. That's what happens when iron oxide nanoparticles accumulate in the tissue. So here I'm showing the result of near-infrared optical imaging because, as you remember, those nanoparticles are also modified with CY5.5 dye. So here you see the accumulation of the nanoparticles in primary tumors and in the lymph nodes and metastatic sites. In this mouse model, it's an MDA-MB231 luciferase expressing, very popular model in breast cancer research. So the next study that we did was actually to test whether injection of our nanoparticle can prevent formation of lymph node metastasis. So here we had mice with primary tumors that were treated with either nanoparticles carrying oligonucleotides inhibiting microRNA 10B or scrambled oligonucleotides that are irrelevant. And as you can see, the animals that were treated with nanoparticles targeting microRNA 10B did not develop lymph node metastasis, whereas animals that were treated with scrambled control went on to developing lymph node metastasis. And this was a very highly reproducible experiment all animals that were treated with this compound that we called MN anti-MR10B, magnetic nanoparticles carrying oligonucleotide to microRNA 10B, they did not develop metastasis. So this experiment showed us that we can deliver our nanoparticles to primary tumors, and they prevent formation of metastasis. Now, if we want to develop an approach where we target already established metastasis, we need to deliver those nanoparticles to distant metastatic sites. And this is what we did in our next study. So here you see an ex vivo, basically excised organs from the animal with metastasis that was also injected with our nanodrug, as we call it. So this is a metastasis, the bioluminescence imaging of lymph node, lungs, bone metastasis. And this is accumulation of the nanodrug. This is a CY5.5 channel, near-infrared optical channel. And you can see there's a nice correlation in accumulation with the bioluminescence expression of luciferase in metastatic sites. So another experiment that we did in vivo, where we showed accumulation of our nanodrug in metastatic sites in the brain. So those are animals with brain metastasis from breast cancer. And you can see there's a pretty nice correlation of accumulation of our nanodrug with bioluminescence signal. So the next question was, can we stop and arrest already existing metastasis? So in this study, we took animals with existing metastasis, metastasized from primary tumors, and we injected these animals with our nanodrug targeting microRNA 10B and scrambled nanodrug. And you can see here that our animals that were injected with active nanodrug arrested the growth of metastasis. You can see here the line is straight. And animals that were injected with control nanodrug went on to not only to developing larger metastasis, but also they spread to contralateral side. And interestingly enough, we also monitored primary tumor, and the growth of primary tumor was not affected in these animals. And this was actually in correspondence with what Bob Weiberg showed, because they showed that microRNA 10B was only responsible for migration and invasion. But what we saw here seemed like it's not only responsible for migration and invasion, but also for proliferation and viability of tumor cells. So we were a little bit puzzled. So to kind of investigate this result, what we did was we took another group of mice with established metastasis, and we excised primary tumor, because development of metastasis is a result of basically two events: repopulation of cells from the primary tumor and proliferation of the cells that are already accumulated. So we eliminated the source of primary tumor, the source of cells that travel to the metastatic sites. And what we saw, and we treated those animals the same way we treated them in this experiment. And what we saw was that the picture was very similar. This was very similar to this, meaning that in the absence of primary tumor, those cells were affected by the nanodrug. Their proliferation and their viability was affected by the nanodrug. So it means that in metastatic sites, the nanodrug action reduces viability of metastatic cells, and not only on the primary site where it's only responsible for migration and invasion. And this discovery had a very significant implication on future treatment of metastatic animals. So in the next study, we decided, because here you can see that we only went up to 4 weeks, and you can say, well, this curve is kind of going up. So what happens after 4 weeks? And you'd be right. So with next experiment, what we did, we took 5 groups of animals, and I know it's a busy slide, and I'll go really fast. So the 5 groups of animals injected with either PBS, scrambled nanodrug plus low dose of doxorubicin, our nanodrug, and then our active nanodrug with a low dose of doxorubicin. Why did we add doxorubicin? Because the cells proliferate very fast in animal models. It doesn't happen in human models. So when we inject our nanodrug, it's actually getting diluted by the proliferation of tumor cells, by the dilution during this proliferation. So it was necessary to slow down the tumor cycle, slow down the proliferation cycle. Hopefully in humans we don't have to do that. But either way, what we saw was that when we applied this mix on the animals with established lymph node metastasis, after only 4 weeks of treatment, we saw the disappearance of metastasis. And you can see that in all those groups, animals continue to develop metastasis. And here, you understand that they just expired. So when we saw the disappearance of metastasis, the treatment was stopped. That's what the red star means. And these animals continue to be metastasis-free for the rest of their experiment and for the rest of their natural life. And here we show the bioluminescence signal, which goes down in those experimental group and actually stays below the baseline. And this is the Kaplan-Meier curve that shows the survival in this group, and the survival was 100%. So now I need to mention that this group of animals was injected with MDAMB-231. It's human cells injected in immunocompromised animals. And of course, we were curious what happens if we have a complete immune system in place. So in the next experiment, we used 41 model, which is injected in immunocompetent mice, BALB/c mice. And this is a very, very aggressive model. After the injection of a primary tumor, we see lung metastasis on the 8th day. Very difficult to treat. So we treated these animals using basically the same treatment groups. And what we see here, that animals with established lung metastasis, these metastases were eliminated about 4 to 6 weeks of treatment. And after they were eliminated, we stopped the treatment, and animal remained metastasis-free for the rest of the experiment. And here you can see the bioluminescence signal, and this is a curve showing the survival. So we achieved a 65% survival, 65% of elimination of cancer in this group, which I think is a significant number. And we continue to work and trying to understand what happened to the other 35%. Though these animals didn't eliminate their metastasis completely, but what was more important, that they didn't spread to the more distant side. They didn't spread to the brain, bone, or liver. So these metastases were arrested. So we think that it's a significant achievement. And in the outlook to clinical translation to the studies, we performed some initial toxicity studies, which are very important, and the part of any therapeutic development. We found that the injection of the nanodrug in combination with low-dose doxorubicin didn't cause any significant toxicity. We did blood chemistries, we did pathologies, and we showed that this is a pretty safe way to treat those drugs, those animals. And when I was still working at Mass General Hospital, but was considering my move to MSU, I got in touch with my future or now present colleagues at the College of Veterinary Medicine to see if we can translate those studies into companion animals, which FDA would require if we want to apply for pre-IND studies. So it was actually a pleasure working with them back when I was in Boston, but now again we continue working together these days. And I just want to show you the picture. This is our first patient, a cat with memory metastatic carcinoma named Pearl. And basically what we tried to do here was not a treatment study because she was at her last kind of days of life, just basically showing that the injection is safe. It's sort of mimicking phase 1 safety studies. So you can see here that she was injected with a drug, and she was active and eating and drinking. And we imaged Pearl, and we showed that there was a significant accumulation of the nanodrug in her metastatic lesions. This is a T2 weighted image. You can see the black dots signifying accumulation of the nanodrugs in her abdominal area. And this is a histology, and the red color here is CY5.5, showing accumulation of the nanoparticles. And we're very thankful to this our first patient and her owner for letting us do these studies. So I also want to mention that we are very thankful to the MSU Foundation who invested in those studies, and hopefully will be able to commercialize them and to bring it into patients. So in my last slide, I just want to show you what I'm here for, why I joined MSU. It's basically the idea of precision nanomedicine and the vision that we have that we want to develop. Just imagine a scenario that the patient is diagnosed with, say, breast cancer, and the patient undergoes biopsy, routine blood draw, and goes through the sequencing of a genome, of tumor genome, and finding abnormalities that can be treated using our nanodrugs. And then we can synthesize this nanodrug that would be treating precisely the abnormalities and mutations in this patient tumor genome, which again goes back to the patient, and all of that could be done under image guidance. So this is the vision that we have. And I just want to mention that while now I'm just talking about microRNA 10B, but this approach can be applied to other microRNAs that are dysregulated in breast cancer, or for that matter, in other diseases where microRNAs are dysregulated, that not only cancer, but other diabetes and cardiac diseases and others, neurological disorders. So, and finally, this last slide, actually, I want to tell you about the vision of precision health program at Michigan State University. So what I told you is just is just a part of it. It's a nanomedicine. It's a precision nanomedicine program. But precision health program at MSU will consist of several components, which would be, first is a healthy body monitoring, because we're switching focus from precision medicine to precision health. We want to see patients more healthy and monitor their health and focus on prevention and prediction. Early detection, as I just mentioned. We're still going to continue working on the molecular basis of disease and develop methods and ways to find various mutations. And one of the most important questions is basically distinguish between aggressive indolent cancers, very important, for example, in prostate cancer, and the mechanism of drug resistance. The therapeutic discovery, we're going to develop this area as well. Like I said, my personal interest is in precision nanomedicine, but there's other areas that we want to develop, such as genome editing and others. We will try to bring all these new developments from bench to bedside, and for that we would need to go through all kind of testing, including PK, PD, toxicity, immune response, and there's an amazing facility in life science building that helps us to do that. And finally, the post-therapeutic era, which is a monitoring of biomarkers of remission and recurrence, and this is something that we're also going to work on. So at the end, I'd like to acknowledge members of my lab at MGH, and some of them moved with me to MSU, and my new colleagues and friends from College of Veterinary Medicine, Eric Shapiro's lab, Department of Radiology, and agencies that fund this research. We have grants from NIH and Breast Cancer Alliance, and I want to thank you so much for inviting me to talk, and thank you for your attention.
So I could do one very short question.
Yes, your research and everything that you've reported on is really wonderful. This conference is a testament to a mixture of various research areas from various places of various foundations of science. Now I'm going to throw in a dead ringer to add another one called economics. What is it going to cost to apply this to individual patients on a massive basis? And I think this should be added to the mixture in everything that's done, eventually to the point of making it affordable. What do you say about that?
Absolutely agree with you. So if we look at the precision health program in general, what we're trying to do actually to reduce the cost of healthcare, because we now know that the later we treat the disease, the more money we spend on that, and the worse the outcome. So what we're trying to do is to treat it early, to detect it early, and treat it with the most efficient drugs as we have. And we believe that the overall cost of the healthcare would go down. In regard to the particular nanodrug that we're developing right now and trying to bring it to the market, we, as I declared by financial interest, we founded a company, Transcode Therapeutics, trying to commercialize this nanodrug for treatment of breast cancer and other metastatic cancers. And we do calculate, of course, how much it's going to cost to do that. And I have to say that as of right now, the cost is actually lower than treating patients with a combination of the drugs that are available now on the market and that are tested in clinical trials.
Okay, let's thank Professor Moore again.